Hello, my name is Deanne Wright, and I'm a member of the SRF Board of Directors and Chair of our Research Committee. It's so great to see all of you here today, and I hope each of you takes away meaningful information from these sessions. Before we begin this session with an introduction to genomic studies, I have just a few quick notes to share with you. If this is your first day of the session, I want to call your attention to the poll on the left side of your screen. This is your chance to share a bit about yourself and learn more about the people in the session with you today. And we do have a live Q&A that will follow the last of these presentations, so please ask your questions. On the left side of your screen, please enter them under the Q&A panel. We'll respond to all questions either here on screen or directly via message. After the Q&A for this three-part genomic session, there will be a 20-minute intermission. And then we'll come back to talk about the SRF's Cure Crew, a grassroots group of volunteers who give their time and talents to help raise awareness and funds for a cure. Finally, this month is Scleroderma Awareness Month, so please join us to help spread the word about this disease. Whenever you share the SRF or scleroderma, you're helping to make a difference in how many people know what scleroderma is and does. Use the hashtag #SayScleroderma and tag us at SRF Cure on Facebook, Instagram, and Twitter. And now it's my pleasure to introduce internationally renowned genomics researcher, Dr. Dan Kastner. Dr. Kastner is the Distinguished and Senior Investigator at the National Human Genome Research Institute, where he runs a large research group. Until just recently, and for over 10 years, he served as the Scientific Director of the Intramural Program at the NHGRI. Dr. Kastner's career has focused on using genetic and genomic strategies to understand inherited disorders of inflammation, and he is responsible for developing the concept of autoinflammatory disorders. Dr. Kastner and his research team have identified the genes responsible for more than 13 single-gene inflammatory disorders, which has led to the identification of currently available treatments for a number of those diseases, immediately helping those patients. In scleroderma, Dr. Kastner has overseen research in both the GRASP project as well as the Juvenile Scleroderma project, both of which you will hear about today. Dr. Kastner is the recipient of many distinguished awards, including the NIH Directors Award, the Paul Klemperer Award of the New York Academy of Medicine, the NIAMS Mentoring Award, and the prestigious Service to America Award. In 2021, he was also awarded the Crawford Prize in Polyarthritis by the Royal Swedish Academy of Sciences. This is considered to be one of the major international science prizes and is also considered to be a complement to the Nobel Prizes. I'm pleased to now turn the presentation over to our dear friend, Dr. Kastner, to give you an introduction to genomic studies.
Hi, I am Dan Kastner. I am a physician scientist at the National Institutes of Health in Bethesda, Maryland, and it is my enormous pleasure to welcome you all to this, the first patient forum for the Scleroderma Research Foundation. And during the next few minutes, I'm going to talk with you a little bit about cutting the knots that tangle us up in terms of understanding human disease with the shears of genomics. And I'm sure that some of you are wondering, well, what exactly is genomics? How does that relate to genetics? And genomics is simply the study of all of our genes rather than individual genes, which would be genetics. And so genomics is a comprehensive approach. And my first encounter with genomics actually occurred some 37 years ago when I was a beginning rheumatology fellow at the National Institutes of Health. And I happened to encounter this patient who had a mystery disease that was characterized by recurrent fevers and episodes of very severe arthritis. It turned out that he had a disease called familial Mediterranean fever, which at that time, back in 1985, was known to be a genetic disease. It was known to be common in people of Mediterranean and Middle Eastern ancestry. It was known to be recessively inherited, meaning that you had to get a susceptibility gene from both sides of the family. But that was about it. We didn't know anything about what the gene was, what chromosome it was on, what that gene did. And it seemed, based on the clinical picture of this patient, that it sure would be great if we knew some of those things. And right at that time was the beginning of the advent of genomics, of the Human Genome Project. And so basically, scientists were beginning to have this idea that we could look comprehensively at all of the genes in the genome in order to figure out the cause of genetic diseases. That was very attractive to me because it is comprehensive. That is to say, we're looking at all of the genes at once. It is unbiased. We're not bringing any presuppositions to the table. It is efficient because we're looking at everything all at once, and it enables diagnosis, it sheds light on disease mechanism, and it provides the basis for new treatments. So sign me up. And that's exactly what happened, is that I got interested in going after the gene for this disease, familial Mediterranean fever. In the summer of 1989, it took a little bit of time to get this organized, I went to Israel and actually collected blood samples on some 62 different families in which there were members who have familial Mediterranean fever and then some members who did not. And then I brought these samples back to the National Institutes of Health, and we began to do the genomic approach. We comprehensively looked at markers on every chromosome in the genome to figure out what chromosome the gene is on. It's chromosome 16. And then we systematically tried to narrow down the region on chromosome 16 where the gene resides, and then to develop maps of that region, and then to figure out all the genes that are encoded in that region, and systematically, one by one, to go through them and try to figure out which one has a mutation associated with familial Mediterranean fever. Actually, it took us some 8 years at that time. The technology was very slow at that time, and we had to invent some of these things for ourselves for that region of chromosome 16. But ultimately, we found a brand new gene that is mutated with this disease. And that, of course, allowed us immediately to be able to do genetic testing to figure out if someone has familial Mediterranean fever, but it also taught us something about the biology of inflammation. It turns out that this new gene is actually the prototype for a whole family of genes that have a particular mutated with this disease, and domain named after the pyrin protein that's mutated in this disease. And you can see here on a model of this pyrin domain that, in fact, you have blue positive charges and red negative charges, and you can have actually interactions between proteins that have these pyrin domains by virtue of these charge interactions. We learned all that just by having discovered this new disease gene. But it doesn't stop there. Actually, we are also able, and we were able to find a new gene in individuals who have something that looks like familial Mediterranean fever, but actually it's not. And in this particular case, a couple of years after we had found the gene for familial Mediterranean fever, looking at patients that have a dominantly inherited disease in which it's not restricted just to people of the Mediterranean and Middle Eastern area, with prolonged episodes who do not respond to the treatment for FMF, we found a new gene that causes a similar kind of disorder, and we call that disease TRAPS. And actually, when that disease was published, you can see the cover of the journal where it was published. Sadly, this is actually a photograph of the autopsy slides from the kidney of the father of one of the patients who we had discovered has this condition. And the father had been treated as if he had familial Mediterranean fever, but in fact, he didn't respond to that treatment. And if we had known then what we know today, we would have treated him differently, and perhaps he would be alive today. In another look-alike disease, a disease called neonatal onset multisystem inflammatory disease, children have fevers from the first day of life. They have a severe skin rash. They have inflammation of the eyes. They have inflammation of the lining around the brain, called meningitis. And they have inflammation of the inner ear that leads to deafness. It's a terrible disease. And actually, the prognosis for that disease is that in young children, usually by the age of adolescence, they will develop blindness, deafness, and intellectual disability, and many of them don't survive into adulthood. But realizing what the gene is that causes this disease, it became clear to us that, in fact, there was a medication that had just been FDA approved for the treatment of rheumatoid arthritis that we could take off the shelf and treat these patients. And when we did that, it was miraculous. These patients had their skin rash resolved, their eye inflammation resolved, their inflammation of the lining around the brain resolved, their inflammation of the inner ear resolved. And patients that we started on this medicine 20 years ago, who are still taking it now, are now graduating from college rather than being perhaps institutionalized or not even surviving. So, what's so great about genomics? Well, it's comprehensive, it's unbiased, it's efficient, and it enables diagnosis, sheds light on disease mechanism, and provides the basis for new treatments. Now, right after we had made that discovery, there was a revolution in DNA sequencing. And basically that allowed for the cost of DNA sequencing to come down dramatically relative to the cost of computing. And that allowed an explosion in terms of the number of diseases that one could solve using the comprehensive approach of genomics. One such disease that we were involved in studying is a disease where children present with early onset of fevers and strokes. We saw our first patient with this, a young child, 2 years old, from New Jersey, who was sent to us with recurrent fevers and stroke. And we had samples from the patient and her parents. And then a few years afterwards, we saw a patient from Texas who, at the age of 6, had had recurrent fevers her whole life and 6 strokes. Using this new sequencing approach, we were able comprehensively to look at all the genes in the genome from both of these children and their parents, and we were able to find a single gene that explained disease in both of these children. And once we did, we were then able to find a large group of children who actually had this same condition. And once we found that, we learned that these kids actually develop strokes on the average of once every 3 years, and there was no treatment for it. But recognizing that they were a group of patients with the same disease, we started to study it. And we, after getting into it a little bit, decided that we would try inhibitors of TNF, tumor necrosis factor, in these children. That had not been tried before. And in point of fact, before these kids went on TNF inhibitors, they had had 15 of them, had had 55 strokes over 2,000 patient months of observation. In the study, we stopped it after 700 patient months of observation because the number of strokes at that point, and still is, 0. So remarkably effective in this disease. But all because we had taken a comprehensive approach and had learned about a new disease in that process. So again, what's so great about genomics? It's comprehensive, it's unbiased, it's efficient, and it enables diagnosis, shedding light on disease mechanism, and providing the basis for new treatments. We've also used genomic approaches to answer the question, how do old people develop inflammatory diseases? For individuals who haven't inherited susceptibility genes, how is it that they develop inflammatory diseases later in life? Well, in order to approach that question, again, we took a comprehensive genomic approach in which we looked at 2,500 individuals with unexplained inflammation and screened them against 841 different genes. And what we found was that there were somatic mutations in a particular gene called UBA1. Somatic mutations means mutations that you weren't born with, but where they occur sometime in development. And it might be from some environmental exposure, such as radiation or some sort of a toxic chemical, or perhaps a virus that one is exposed to, something in the environment. And some of the cells in the body have the mutation, and some of the cells in the body don't. And if you look at this slide, one can see that in the blood of these patients, they have both the normal in red and the mutant form of this particular gene. But if you break it down, if you look at the blood cells according to their cell type, you can see that some of the cells actually have the mutation, as shown here, and others of the cells do not have the mutation. That's exactly what one would see with somatic mutation. Well, in fact, if one uses that insight then, and you look at patients that develop inflammatory diseases later in life, you see that patients that carry a whole bunch of different clinical diagnoses, they all have somatic mutations in this gene. It's not like everybody with those diseases does, but a subset of them do. And because of that, we group these individuals together, and we call that a new disease that we call VEXAS. And VEXAS actually has a number of manifestations that some of the patients didn't even know they had before we knew to look for it. They can have inflammation in the lungs. They can have white blood cells, inflammatory white blood cells that get in the air exchange areas of the lungs. They can have inflammation of the blood vessels. They can have inflammation of the ears. And they can have these funny looking bubble-like structures in some of the cells in the bone marrow. So in any case, what's so great about genomics? It's comprehensive, it's unbiased, it's efficient, and it enables diagnosis, shedding light on disease mechanism and providing the basis for new treatments. So the last question we have is, what is the genetic mechanism of common inflammatory conditions? And we approach that studying a disease called Behçet's disease, in which patients have multiple painful ulcers in the mouth. They have inflammation of the eyes. They have ulcers in the genital areas. They can develop pustules on the skin when you prick their skin with a sterile needle. And we have looked with various genomic approaches, comprehensive approaches, to see that in a few cases, there's a single gene that can cause this disease. But in a lot of cases, it's actually the combination of a whole bunch of different genes, each of which contributes a little bit. And you can have almost like a Chinese menu kind of situation in which various combinations of those genes will lead to Behçet's disease. Well, interestingly, some of those very same genes have recently been shown to cause just canker sores in people that don't have Behçet's disease or any other inflammatory condition. And we have looked at a condition called FAPA syndrome, which is a syndrome in children in which they get sores in the mouth, sore throat, and swelling of the lymph nodes in the neck. And again, the same genes that are involved in Behçet's disease and canker sores can predispose to this condition too, making us think that in fact there's a spectrum of disease. We call it the Behçet spectrum disorders that involve these variants in these particular genes. What's important about that is actually, though, that treatment sometimes can be suggested by this. And in fact, in these kids that have this periodic fever syndrome, that in fact, a treatment that had just been FDA approved for Behçet's disease, we're now finding to be effective for some kids that have that condition. So in any case, I think that it's evident that in fact, genomics can teach us a lot. And in the 37 years that I have been doing this work, genomic analysis of human diseases, I can say that it is exhilarating, it is gratifying, and that there are two key ingredients in doing this. One of them is patience. And that's why some of you are here watching this, is to learn what patients can do in collaboration with the Scleroderma Research Foundation. Patients are essential to these kinds of studies. And then the other thing that's essential, patience with the CE at the end, that is to say, it does take some time in order to get all of this done. So at this point, I'm now going to turn things over to two colleagues who are funded by the Scleroderma Research Foundation and whom I am fortunate enough to be collaborating with. And they are going to tell you about how the shears of genomics can be used to study scleroderma. Thank you very much.
Thank you, Dr. Kastner, for that illuminating introduction to genomics research. For today's participants, we are going to hold questions until the end of all of the sessions on genomics, when we will be able to have a group discussion. Now, I'd like to introduce our next two speakers, Dr. Francesco Boin and Dr. Pravitt Gourh, who will be sharing their work on the GRASP project, which stands for Genome Research in African American Scleroderma Patients. I want to mention that the Scleroderma Research Foundation has been a partner in the GRASP project since its inception, and this project is an example of how nonprofits like the SRF can partner with the NIH to fund important scleroderma research. From this talk, you will learn why SRF researchers believe that studying African American scleroderma patients is important, what has been learned from the project, and how GRASP is helping to understand scleroderma in the African American population, as well as in patients of other ethnicities. Dr. Francesco Boin is the director of the Rheumatology Division and a member of the Kao Autoimmunity Institute at Cedars-Sinai in Los Angeles, California. Prior to his work at Cedars-Sinai, he was the director of the Translational Research Program at the Johns Hopkins Scleroderma Center for a decade before joining the University of California, San Francisco in 2015 and establishing the UCSF Scleroderma Center. His clinical activity is devoted to patients with fibrosing skin disorders such as scleroderma, as well as primary Raynaud's phenomenon and other systemic rheumatic diseases. Together with Dr. Fred Wigley at Johns Hopkins, Dr. Boin has led the clinical side of the GRASP project, coordinating the large consortium of clinical centers that enrolls patients into the study and the collection of data and patient samples, and importantly, in the analysis of the clinical data. Outside of the GRASP project, Dr. Boin's research focuses on understanding the immune response that drives autoimmunity in scleroderma. Joining Dr. Boin will be Dr. Pravitt Gourh, who is a board-certified rheumatologist and clinician scientist with a long-standing interest in scleroderma. Dr. Gourh currently directs a translational research program at the NIH that is focused on understanding the genetics of both pediatric and adult scleroderma, biomarker discovery, and disease classification. He is an assistant clinical investigator at NIAMS. Dr. Gourh has been a critical member of the GRASP project and has been a leader in analyzing the genomic data generated by the project, as well as in the subsequent studies that aim to understand how genomic differences can lead to scleroderma. And now, Dr. Boin and Dr. Gourh.
Good morning, everybody. Again, thank you very much for the kind introduction. It's a pleasure to be here from the Cedars-Sinai Medical Center and the Kao Autoimmunity Institute in Los Angeles. Today, I will briefly introduce you the GRASP project. So what is GRASP? So GRASP stands for Genome Research in African American Scleroderma Patients. And basically, this is a consortium of investigators with clinical and research expertise in scleroderma that gather from more than 20 academic centers across the United States with the goal of studying the genetic aspects of scleroderma in African American patients. The group started working together back in 2012 under the leadership of Dr. Wigley from Johns Hopkins, Dr. Gourh, and Dr. Kastner at the NIH, myself currently at Cedars-Sinai. And importantly, the Scleroderma Research Foundation has been involved since the very beginning, being instrumental to bring all these investigators together, supporting all the activities that GRASP developed over the past several years. Now, why studying scleroderma in African American patients? And why do we need a consortium of expert centers to do so? Well, to answer to this question, I think we need to look at how scleroderma affect our population. Scleroderma is a rare disease. In terms of prevalence, so total number of cases, In the United States, it's about 300 per million population. Incidence, which means new cases per year, it's about 20 cases per million population per year. So compared to the rest of the world, we are on the high end of the spectrum. It appears that North America and the United States have higher numbers and more frequent incidence and prevalence of scleroderma. Now, what it means is that in the United States, there are about 100,000 Americans suffering from scleroderma. And every year we have several thousand, 7,000, 8,000 new cases that are diagnosed or that develop. And I think this is important because while it is a rare disease, it really hurts people. It really matters given its high morbidity and mortality. If you think in terms of large urban areas like New York or Los Angeles, there are several thousand of patients out there, and many hundreds that every year develop the disease. And if you think how many specialized centers, make me realize that certainly we don't have enough expert places to develop and deliver the best care for our patients. So certainly it's an area that we want to focus more. Now, if you were to look at scleroderma patient from a distance, let's say from a satellite that looks down to see scleroderma patient, they will more or less look the same, like little blue dots scattered throughout the United States. But we learned that scleroderma patients are not all the same. If you were to look close enough, perhaps using a special lens, you will discover that every patient is unique, is different. And actually, some patients are more similar to others, and you can identify different subgroup of patients based on different characteristics like gender, demographics, ethnicity, and so forth. So I think the development of specialized center and this center working together generate really the possibility to look at large scale this patient population and start to identify these different clusters. Let's see some examples. So if we use the lens of gender, for example, the female to male ratio is 4:1. We know that scleroderma is more common in women compared to men. But then when we look at the characteristics in female versus men, we realize that actually in men, the disease is worse. And I'll give you an example. This is the Pittsburgh cohort, large cohort of almost 3,000 patients. And they look over 10 years, the probability of survival, so the mortality, and independently, male gender was associated with poorer survival. Let's look at the age of onset, for example. This is another large cohort. This is the Johns Hopkins cohort, other several thousand patients. And look at the distribution of this bar. You see that typically scleroderma, this is when the scleroderma typically starts here in the center in the mid-40s. But we know that there are cases with early and cases with late onset. And actually, when we look at the characteristic of the disease, it looks like that early or late onsets have different type of scleroderma, and possibly more severe, more extreme. So this is very important because then we ask the question, how about racial differences in scleroderma? So reflecting the distribution of race in the United States, certainly Caucasian is the largest group of scleroderma patients that we normally follow in our clinics. But there is no doubt that any other ethnic subset are affected as well. And so the big question that we have is, are they affected the same? Is every ethnic group affected the same way? And we know the disease don't treat everybody the same way. So I'll take an example. This is a recent one, the one that we all have been concerned about. We look at COVID mortality rate in the United States. This is recent data. Throughout the pandemic, African American or Latino were almost twice as likely to die from COVID compared to white or Asian population. So clearly there is a disparity. And when we look about scleroderma, well, that's one of the main reasons we wanted to generate GRASP, to do a study focused on African American, because from our clinic, we realized that African Americans have higher incidence, earlier onset of the disease, more aggressive, more diffuse skin with vascular manifestation, more lung involvement, and ultimately decreased survival. So let me give you an example here. This is a study from Michigan where they look at scleroderma incidence by Asian race in women. And as you can see, the blue line, tend to be higher than the red dotted line. And so the blue line is Black female. They have much higher incidence and they peak earlier. So that means that it's earlier onset and more frequent compared to white women. Or let's look at mortality. Another big study here from the University of Pittsburgh. They look at risk factors for mortality within 5 years from first visit. And then they look at it independently, just the race, in this case, African American, was giving them more than 1.5 chances of dying in 5 years from the diagnosis. Race itself was a significant risk factors. And if you look again at the population-based study, this is all America for 48 years looking at death, you can see that compared to the early 70s, there is a better, so there's been a steady increase of mortality, and then after 2000 start to get better. But the gap between Black and white, it was not filled. Still there is a discrepancy, there is a disproportion in terms of number of deaths. So no doubt that clearly the understanding the higher disease burden measured in terms of incidence, severity, mortality in African American is an unmet need. And we were also thinking there is another gap, there is another disparity. And the fact was that main studies investigating the biology and specifically the genetic of scleroderma susceptibility was mainly in the literature, in the medical literature, is mainly focused on Caucasian. There was not much on African American. So we really felt it was clearly important to really start to address this problem. Now, why are there disparities in outcomes in African American scleroderma patients? There are many possible reasons. Some are, socioeconomic status, access or quality of healthcare, raising the question about inequity and equality, and these are important topics to be addressed. Cultural differences, the environment, different diet, and certainly different biological difference. The genetic background may have an impact on how the disease manifests. And so clearly, we wanted to leverage novel technologies and the growing sophistication of computational tools to start understanding how biology, in this particular case, how the genetics contributes to health disparities, and how these biological factors may actually interact with the other elements that are critical in determining outcome. So what are the main objectives of GRASP? So first was to collect clinical data and DNA samples from African American patients as well as from healthy controls. And so clearly that's why we needed a consortium. That's why we needed people representing centers that cares for scleroderma patients that have access to African American population across the United States. And we succeeded assembling really the largest national cohort that has ever been assembled of almost 1,400 scleroderma patients. And in collaboration with the NIH, we were able to have more than 1,000 healthy controls to compare the genetics and understanding what's unique about the African American patients. The second major goal was to establish a well-organized and comprehensive clinical database and sample repository. And I think, I'm really grateful. I've been very lucky to have great collaborators in the leadership of the GRASP study, developing the necessary infrastructure to keep momentum in our work. So we had Johns Hopkins being the database center, coordinating center, the NIH, receiving all the samples from all over the United States and becoming the experimental hub. And certainly on the background, the Scleroderma Research Foundation, I'm very grateful to Deanne Wright as just being always an amazing support and an enthusiastic partner throughout this journey. And then the third goal, obviously, was to perform the most advanced cutting-edge genetic studies. And what we mean for that is that we wanted to investigate the variation of gene sequence, how the sequence of the genes across the entire genome, to try to understand if there are changes in the genes, small changes that explain why in African American scleroderma has higher frequency, severity, and mortality. And ultimately, to benefit every scleroderma patient. We do believe that unraveling the secret of how scleroderma create higher burdening in a smaller group, such as African American, can shed light and help us to care more and better also for every other scleroderma patient of other ethnicities across the world. So I conclude here. This is a picture from our last meeting. We always have great time and wonderful memories about all the investigators were getting together. And so I really thank them, Academic Center, Hospital, the NIH, and the SRF, who really participated so generously to this wonderful partnership and journey. And now I pass the presentation to Dr. Gourh, who will talk about the genetic studies that we perform and some important initial findings. Thank you.
Thank you, Francesco, for describing the clinical phenotype of scleroderma and introducing the GRASS project, that is the Genome Research in African American Scleroderma Patients. I am Dr. Pravitt Gourh, and I'm a rheumatologist and a physician scientist at the National Institute of Arthritis and Musculoskeletal and Skin Diseases at NIH. Today, I'd like to tell you a little bit about what we have been doing in the lab with this wonderful collaboration GRASS that has been established. So before we get into that, let me just tell you, why should we study genetics and what is genetics? So studying genetics using patient samples is especially useful in understanding human diseases. Our bodies are made up of tissues, and these tissues are made up of cells. And in each of these cells is, as you can see right here, is DNA. Now, DNA, you can think about it as like a cookbook, and each chapter in this DNA is a recipe. And each of this recipe makes a certain dish, or what we call as a protein. So if we can figure out or identify misspellings in the recipe or the gene, then that can tell us what went wrong in the dish or the protein, and can help us better understand the human disease. So studying genetics or studying these misspellings in the DNA can give us a better idea on why some people might be getting scleroderma, and also what can we do about it? How can we better develop a drug to specifically go after that protein and fix it? So our goal has been to study the DNA of scleroderma patients to identify these misspellings and to help come up with newer treatment options. So this is my lab, and my lab has focused on studying scleroderma in the African American population. And the reason for that is, as Francesco was telling you, African American patients have a higher disease burden than European ancestry patients or Caucasian patients. The disease starts at a much younger age, an earlier age. There's a higher prevalence of scleroderma in the African American population. So in the United States, in red as I've shown you here, the prevalence or the presence of scleroderma is about 315 individuals per million, which is about 2.5-fold higher than the Caucasian population. And if you compare to Europe, it's about less than 100 cases per million. And if you look in East Asia, it's less than 50 cases per million. So definitely, the African American population has a much higher prevalence of this disease. And we are trying to understand why that is the case. Also, as Francesco was telling you, there's increased prevalence of lung involvement or lung fibrosis, which is the biggest cause of mortality or death in our scleroderma population patients. Also, from a genetic standpoint, the African American population have a higher genetic risk. Now, I don't want you to get confused by this, but the point of this statement is that the African American patients have something called a lambda S value that we calculate in genetics of 26, whereas the Caucasian patients has about 12. So it's again almost 2.5-fold higher than the Caucasian patients, suggesting there's a higher risk of misspellings in the DNA that are being carried that might be causing the disease. So what is our hypothesis? So we are suggesting that there are misspellings in the DNA that are of African ancestry that are present in our African American patients. And when they're being carried over or being inherited, they increase the prevalence or the presence of this disease and also this phenotype that is seen in these patients. And if we can identify these, then we can identify these genes and perhaps target them and come up with treatment options to help the patients. So we have taken a multifold genetic approach, but before we get into that, we established this wonderful collaboration of 26 centers of different universities all around the United States and collaborating with NIH right here to figure out this disease. So we've taken a sequencing approach and a genotyping approach. I don't want you to go into details about this, but what I want you to know is that these are latest cutting edge technologies that we are utilizing and implementing to help figure out this disease. So now here is some of our results, and it's a little bit complicated and confusing. So let me just tell you, I was telling you about the misspellings in the DNA. So each dot or the circle that you see here is a misspelling in the DNA. And the taller or the higher it goes, this line, the more likely that it's causing an increase or is associated with the disease, in this case, scleroderma, and increasing the risk for the disease. So now I want you to focus on this green box right here. And these orange dots that you see here are misspellings in this gene. And we feel we have shown in the lab that they are affecting this protein called TGA-beta3. Now, what's important about this is this, that these misspellings are only seen in the African population, not in any other population, number one. Number two, this gene, TGA-beta3, is being reported for the first time with African American scleroderma patients, never reported in Caucasian scleroderma or any other disease. So now this is a new target that could potentially be a game changer for patients. The other thing I want you to show and talk about today is this other peak. This is the tallest peak. These misspellings are in genes called HLA genes, which are responsible in our immune response. And a lot of diseases that are autoimmune have misspellings in these genes. But this is where I want to focus today. A couple of years ago, we published a paper in Proceedings of the National Academy of Sciences and identified two misspellings, one right here and the other one right here, that are increasing risk for scleroderma about threefold or so. And both of them are African ancestry specific again. That's what we wanted to identify. Variants or misspellings in the DNA that are from African ancestry that are being present in our patients. So when we look at how common are they in our patients, so they are seen about in 24% of our patients who are African American, as compared to only 9% of healthy individuals. Now, if you compare them with Caucasian population, they have 10-fold lower frequency, very little frequency of this misspelling. Even in Europe, it's less than 1%. And if you look at in East Asia, it's again very low frequency. But when you look at the continental Africa frequency and you look at the Nigerian population, which is the ancestral population for African Americans, it's 12%. That's still less than half of what we see in our patient population, suggesting that carrying of this misspelling is perhaps increasing the risk for this disease. So what can we do with this information and how can this be helpful to the patient? So let me give an example. So some of you may be familiar with these antibodies that are present in scleroderma, and one of them, especially common in African American patients, is fibrillary antibody. So if, say, we have patient number one with fibrillary antibody and who carries this misspelling in their DNA, and we have calculated the risk for lifetime risk for getting fibrosis of the lung is 0.4. So it's a low risk. Whereas there's patient number two who has this antibody, topoisomerase antibody, and they have this other misspelling in the DNA, and their risk for lung fibrosis is 3.7, much higher than this. Actually, it's 9.3-fold higher in patient number two as compared to patient number one for getting lung fibrosis. Now, this information is very interesting for a couple of reasons. Number one, both these tests can be done at any time, especially including day number one of your clinic visit. Just a simple blood test, and we can get this information and can come up with an estimate. What is the likelihood you're going to get lung fibrosis or not? And that can be very helpful because we can start closer monitoring of the patient. We can see them in clinic more frequently, which could lead to earlier diagnoses. We can start the treatment for the lung fibrosis early on, and overall will lead to a better outcome. So that's number one. The other thing how this can be useful is, can we predict lung fibrosis even before that in some people? So we use different computer predicting technologies called machine learning methods. And we use the autoantibodies, or these antibodies like topoisomerase, fibrillary, centromere that you may have heard of. And we use the misspellings in the HLA gene and misspellings in the T cell gene, and they're involved in the immune response. And I'm not going to go into why we chose these, but when we use these, we have about 88% predictive accuracy. That means 88 times out of 100, we can tell if this person is going to get lung fibrosis in the whole lifetime or not. And I think that's pretty amazing. And this is the direction we are trying to take with doing these genetic studies and making a difference by identifying new targets and being able to predict the disease early on. So I hope I've been able to convince you that genetic studies are really important, and studying these diseases using human samples is absolutely critical. And this will give us more information about scleroderma, and we can identify these different subtypes of the disease, whether they are based on antibodies or based on these misspellings of the DNA, which can eventually lead to new drug targets that we have. And finally, all of this wonderful work could not have been done without the contribution of all the patients that we have. And we have over 1,300 patients so far enrolled in this study. And other collaborators and our funders, including NIH and Scleroderma Research Foundation. So thank you so much for your time, and I'll be happy to take any questions at the end. Thank you, Francesco.
Dr. Boin and Dr. Gourh, thank you both for walking us through the nuances of the GRASP project. For our final segment of this genomics session, we will learn about juvenile scleroderma and genomics from Dr. Catherine Torok. Like the GRASP project, the SRF has been a partner in the project to study genomics in juvenile scleroderma since its inception. And we are very, very excited about the potential for this project. Please stay tuned after this session for the genomics Q&A. Today, I'd like to introduce Dr. Catherine Torok, who is the director of the Pediatric Scleroderma Clinic at UPMC Children's Hospital in Pittsburgh. Dr. Torok juggles seeing patients in the clinic, conducting her own research to help develop more effective therapies for localized and systemic scleroderma in children, and mentoring the next generation of pediatric rheumatologists at the University of Pittsburgh School of Medicine. She is the principal investigator of the National Registry of Child Onset Scleroderma, where she collects clinical assessments together with blood and skin biopsies to investigate key molecules promoting scleroderma in kids. She has also been a principal investigator of the Juvenile Scleroderma Project, a project aimed at understanding genomic differences that may contribute to pediatric onset scleroderma, and that's the project you'll hear about next. Here's Dr. Torok.
Thank you, Deanne, for that introduction. I appreciate it. And thank you to everyone for listening. This is Catherine Torok. I'm a pediatric rheumatologist at the Children's Hospital of Pittsburgh at the Scleroderma Center in Pittsburgh. Listed are my collaborators for this project that I work in tandem with the Scleroderma Research Foundation, and Stephen, Elaine Remmers, and Hiro Oda. So we're going to talk a little bit about what scleroderma is in the context of children. This might be different from some of the other webinars you've been listening to. So scleroderma for everyone means hard skin, and it's an autoimmune disease. There are two types. In pediatrics, we actually focus on both. Systemic sclerosis, where most of you are familiar, and localized scleroderma. I will give some examples of both in a minute. So they have different clinical features. Therefore, they are distinct diseases. However, there are some common disease problems and mechanisms. Some things that are often the immune system and the collagen that are shared between the two. But one does not evolve into the other. A lot of my patients ask me that question, so I just wanted to say that up front there. So what's going on? So this is an autoimmune disease. So the immune system that's supposed to protect us from germs, bacteria, and viruses is instead triggering inflammation that helps kill that germs, but then that inflammation stays on. That signal does not turn off, and it starts to recognize the patient's own skin, the own fat underneath the skin and their own organs as foreign. So it starts attacking them and causing inflammation in these organs, including the skin, the blood vessels, etc. So this overactive immune system in scleroderma then starts causing some other tissue changes. So in the skin, the collagen is sitting there and it gets really upregulated, thick, etc., and that's what causes that hard skin. This also occurs in some of the internal organs in systemic sclerosis. The blood vessels also get affected in a similar way with both inflammation and then also the fibrosis. So again, I'm going to talk to you about localized scleroderma and systemic sclerosis because those are important in children. Then we're going to talk about the genetics behind it. These two are similar because of the collagen connection. They both have the abnormal thick collagen or fibrotic skin. So this is just a histology picture. So if you ever had a skin biopsy, the pathologist looks under the microscope and this is what your skin looks like. Healthy skin has some different layers, maybe one or two immune cells here or there, but not that many. Where in scleroderma skin, whether it's systemic sclerosis or localized scleroderma, it looks exactly the same to the pathologist. They would have no clue which disease process they're actually looking at because both have this increase in immune cells or lymphocytes around the blood vessels in the deeper layers of the skin or the dermis or reticular or deep dermis. Later, this inflammation causes that collagen deposition that most of you are familiar with, that thick skin, whether it's localized or systemic, both occur. So we're going to talk about how genes might be interesting and driving this immune system, this inflammation of these cells into the tissue that shouldn't be there. So that's why we're kind of giving you some background here, because I believe it's this inflammation that's driving the fibrosis. So we want to get to those immune cells that are dysregulated and then immune genes that are off. So again, a little just background, systemic sclerosis, you guys are familiar, skin involvement, pretty much widespread. Raynaud's is highly associated, and there's lots of joints, muscle, lung, kidney, et cetera, involvement. Whereas localized scleroderma, I'm studying this as well. We're going to talk about the genetics as well in localized scleroderma because in pediatrics, this is much more common than systemic. So I just have access to both in my registry, and I just have more patients with localized. So I'm kind of a little bit ready at the fingertips there. But it's about 8 to 10 more times common in children, the localized scleroderma. And the skin does look similar in certain aspects where it can be thick and bound down, but typically, unilateral band or line down like an arm or leg or face. And there are other or non-skin extracutaneous manifestations like joint contractures. If it's in the face, they can have some brain, eye, or oral changes as well. But it just doesn't affect the kidneys or the lung or the GI system like systemic sclerosis does. So these are pictures. They're always easier than just me telling you words of what things looks like. So these are all children that I see in my registry with juvenile onset systemic sclerosis. So like adults, you can have a high skin score with that bound down skin throughout, decreased range of motion of the hands or fingers, nail fold capillary changes, which goes along with Raynaud's in a little 2-year-old. This is what it looked like. Some facial changes that look similar to adults with a tight face and thin lip. Some interstitial lung disease. This is a little 7-year-old. Digital ulcers and telangiectasia. So these are same findings you see in adults, but I'm just showing you a pediatric presentation. And why we're interested in the genetics is because these kids are getting it early. This is, 8 years old is the average age of onset. Let's go to the genes. There might be more of a genetic imprint in these kiddos compared to your average adult patient where the onset's around 40 years old or so. These are pictures of localized scleroderma, again, just to familiarize yourself with it. This is linear scleroderma of the leg. You can see you can get a significant leg length discrepancy. This young lady actually just had orthopedic surgeon to break and correct her leg with the lengthening rods because she was having such problems with hip pain, et cetera, because they're almost a 3-inch difference. Here's another linear of the leg, linear of the arm where there's a decreased range of motion, and then linear of the face, sometimes called en coupe de sabre or sort of the saber, and Perry-Romberg syndrome where half of the face is smaller. Not uncommonly, 30 to 40%, they will have brain MRI changes that are actually in that same line where the skin changes are. So just bringing this to people's attention, it's not just a skin disease. And this is kind of going over that both in juvenile localized and systemic sclerosis, the average age of onset is around 8 years old. So both have a relatively young onset. And sometimes there's cases where they're almost congenital, not pure congenital, but like very young, 9 months old or so. But the majority tend to be around school age. There's a girl, more female predisposition, but the ratio is different than adults. It's a little more even, 2-to-1 to 4-to-1 ratio, females to males, whereas in adult systemic sclerosis, it's more 9 females to 1 male. So a little hormones may or may not play a role here. That's what we're trying to find out with the genetics as well. And then we need to get more information, but thus far it seems like juvenile localized scleroderma is more common in Caucasians. We're not sure about systemic because it is so rare. We do see it just across all races. And then, so why are these children getting scleroderma? We know from scleroderma, lupus, dermatomyositis, all autoimmune diseases in general, there's some dysregulation of the immune system combined with environmental exposure and immune dysregulation in the genetic background that make the perfect storm of an autoimmune disease. What's special about localized scleroderma, scleroderma in general, is that there's also a fibrotic component with the fibroblasts and the vascular component with the endothelial cells. So this study that we're going to talk about now is focusing on the genetic component contribution to the disease pathogenesis. So what makes the cells misbehave? I'm a pediatrician, so I got to throw in some fun slides here. So genes, so they're the genetic code that code initially, and then things go downstream from RNA, et cetera, down to protein level to make the genes kind of turn on or off or kind of read a little bit different than they should be. So our initial genetic findings were actually a collaborative group between Pittsburgh, including myself and Dr. Metzger, and Stevens and colleagues at Seattle, and then Maureen Mays and colleagues down in Texas of 76 children. And as you probably heard Pravitt talk about HLA findings in adult systemic sclerosis, we looked for those. We found a few, but not the classic ones to be that predominant in juvenile systemic, so there is some difference. We did see one of the ones that's associated with adult onset systemic sclerosis be associated in children, but we did find some novel associations like DRB1 star 10 to be different in juvenile systemic sclerosis compared to adults. And they did correlate to age of onset. There's some HLA findings that were associated with like 5 to 9-year-old onset versus the older preteen and teen onset, and associated with autoantibodies like topoisomerase and centromere. So very interesting initial findings, and that led to this current collaborative opportunity with the Scleroderma Research Foundation, kind of leveraging existing genomic experience from well-known Dr. Kastner and his pipeline of genetic discovery at the NIH. So basically collaborating with Dr. Kastner's lab, and then I have a very large pediatric scleroderma registry called the National Registry for Childhood Onset Scleroderma, and my colleague Ann Stevens holds a similar registry and repository at Seattle. We kind of came together to put this proposal together for whole exome sequencing now that this technology is available and reasonably affordable to look for mutations in the immune system in pediatric scleroderma. So this is the overall plan. Basically, we want to do whole exome sequencing, and we're doing it from kids. Initially, it was juvenile systemic sclerosis, and we expanded to juvenile localized scleroderma. The juvenile localized scleroderma, though, focusing on 8 or younger at the onset, just because it is more common. We want to get the kind of younger kids that are affected. And we're doing this in a trios grouping. That means you get a little bit more bang for your buck when you get like DNA from mom, dad, and the child, and you see what's different in the child compared to mom and dad in regards to their immune system genes. So this is not 23andMe or anything like that, but it's specifically focused on the immune system genes like T cells and inflammatory markers, cytokine genes, things like that. So we collect that from the patients, their parents, and if available, siblings and grandparents. And then we have a second wing of the study once we find some candidate genes to do it on patients that are individuals and not necessarily the whole family. But then we find the genes of interest by doing that. And at the same time, we're going to biobank the blood from these children because they're in our registries. They're getting blood drawn for routine clinical purposes. We asked to get a couple extra for cells, sera, plasma, RNA, and DNA for future studies. And everyone is usually more than willing to do this because they want to help not just themselves, they really want to help the next generation of kids that have scleroderma. So this is the design. Sorry, it's a little bit busy, but basically we're getting spit from the patient and the mom and dad. And we're expecting in that trios to get maybe 15 to 30 genes that are variant in the child compared to the mom and dad regarding T cells, et cetera, immune cells. And then we look at about 50 to maybe 70 other kids with localized scleroderma, and then also in a separate group, kids with systemic sclerosis. And then we're guessing that between, those groups of patients, we'll probably find about 2 to 5 pure candidate genes that we really think are doing something, that they're really related to the pathogenesis of scleroderma. Then the next step is, once we identify those genes, to start to look at them in a validation cohort, so a separate group of scleroderma patients that we kind of have planned out, and then also recruiting new, so we're welcome for more friends. And then also mechanistic studies, looking in kind of fibroblast tissue cultures, cellular flow experiments, et cetera, to see, okay, are those genes really doing something? They may be different from mom and dad, but are they doing something? So that's kind of the next step of this program. So this is kind of listed out in verbiage, what I've already said, but this is the research team I mentioned. My coordinator is Haley, and that's how to reach us if you're interested. I'll mention that at the end as well, the scleroderma at chp.edu. And so far, we've got 286 samples with whole exome sequencing through the NIH sequencing core, which is awesome. Really excited about this. So basically 39 individual juvenile systemic sclerosis patients, with their families. So the 2 parents and then a couple extra siblings and grandparents, et cetera, and 42 individual juvenile localized scleroderma with their mom and dad, et cetera. So this is really exciting for us. We have additional 13 individual patients with their parents with localized, and 6 individual juvenile systemic with their patients, with their parents in our lab, ready to send to the NIH, and we are open for more. So far, basically, this is the first whole exome sequencing, and especially in the trios format, which gives us more information to date in pediatric scleroderma, both localized and systemic. So we're really excited about it. We have found 17 candidate genes. We're really excited. They're in 3 main pathways. 2 pathways are more of the inflammation pathways, and then 1 pathway is more of the collagen dysregulation pathway. The only caveat is we've only found 1 individual patient affected with each of these interesting candidate genes that we think might be doing something. So we need more patients. We really need like 2 to 3 patients with the same gene variant to really feel more confident that that might be propagating scleroderma in kids, and then maybe in adults in the population at large. So we are working on testing some individual patients that don't have the mom and dad right now on these 17 candidate genes with the NIH. But again, we're happy to grab more patients. And then we're looking at some of the skin biopsies that we've done and looking at these genes in the skin and seeing if they're dysregulated. And we are seeing some pathways kind of matching up with some of these genes. That's really exciting too. And then we're doing some flow experiments with the peripheral blood cells from the patients, as I mentioned, that we kind of biobanked over time. So thank you to everybody. This is a huge effort, huge, between multiple institutions and lots of scientists putting some effort into figuring out some of these more mechanistic studies of these candidate genes. And obviously our patients could not do anything without our patients. And it takes a lot to, spit in the kid, mail it back to us, track down mom and dad and everything, to do a couple little questionnaires. But the patients are really engaged, the parents are engaged, so we're really excited. So thank you to all of those who have contributed, and we're excited for those who want to join us. My coordinator's information there is just scleroderma at chp.edu, and just mention TRIO study and that you're interested. We'll chat more, but we can just send you a spit kit, things of that nature. And then I also, a big shout out, obviously, to the Scleroderma Research Foundation. They were the ones in the spring of 2020 that really chose our project as a research challenge project. So that was really exciting and brought a lot of attention to this study. So I just want to say thank you to Deanne and Luke and the SRF Advisory Board. So Thank you.
Well, thank you all for being here for the Q&A today, and thank you for those wonderful presentations. I thought I would start off with a question for Dr. Kastner about how genomic studies in complex diseases like scleroderma differ from the kinds of studies you described, such as the study you did in familial Mediterranean fever. And why does something like the GRASP study have to enroll so many patients?
Well, Deanne, thank you so much for that question. And I do want to thank the Scleroderma Research Foundation for putting together this wonderful session today. So in contrasting the diseases in which there's a single gene that causes the disease versus some of the more common disorders, one has to bear in mind that for these single gene diseases like familial Mediterranean fever, that you have one gene that is having really a major effect. And so you can actually trace that effect through the pedigree of a family. And so that makes it so that at least if you have the right families, you can sometimes use just a very small number of family samples in order to discover the gene that's involved, because it's basically one gene that's having a big effect. But the way it works for the common diseases like scleroderma, or at least not that scleroderma is real common, but at least the more common diseases like scleroderma, is that in a lot of cases you have multiple genes that are contributing just a little bit to the susceptibility, and you have to combine together those effects of the many genes that are contributing. And in some cases it's tens, sometimes it's hundreds or even thousands of genes that may be contributing, each one just a little bit. And because of that, you can't really trace the effect of one of those genes through a family. And so instead, and in fact, you seldom see families where you have multiple people in the family with the disease, or at least that are closely related to one another. And so the structure is quite different. You don't have the families. Instead, you have a number of people who have scleroderma unrelated to one another, and a number of people who don't have scleroderma, and then you're comparing them, the affected to the unaffected people, and you need lots and lots of subjects in order to see those little effects. And so that's why it takes so many subjects enrolling in a study like GRASP.
That's great. Thank you. Dr. Boin, let me ask you a little bit about the GRASP project, which studies African American scleroderma patients. So you, together with Dr. Fred Wigley, really led the immense effort to organize this cohort, the largest ever of African American patients. So what are the most important things that you think we've learned clinically from the organization of the cohort and the initial analysis of clinical data?
Yeah, thank you, Deanne. It's great to be here. So first of all, as you emphasized, this is definitely by far the largest cohort of African-American patient ever assembled. And this is in itself a great accomplishment because it allows us to really look in a granular way to the clinical features of these patients. The second major point is that they represent clinics and different areas of the United States. So for the first time, we don't have only a specialized center who is studying African American scleroderma patient, but we have a good snapshot across the United States. And this large cohort allow us to confirm what smaller studies have suggested, that the burden, the clinical burden of disease in African American is much more severe, much more progressive and limited in scleroderma patients, in African American patients. We learned that they are more keen to develop aggressive lung disease. We learned that even the vascular manifestations are more severe, and I think this really raised a new awareness for everybody that participating in GRASP on how we approach different patients of different ethnicities in clinic. This was just the beginning of an awareness that now I think will translate into a different way of managing these patients.
So the collective knowledge within GRASP has increased with these studies, but also the information is disseminated outside of GRASP.
Absolutely. Indeed, we started to publish finally after all the effort that was Dr. Wigley, but Dr. Gourh and Dr. Kastner really completed the work of the experimental part. So now the data start to be published, and this is public common knowledge. And I want to emphasize that the consortium is more than 24 academic centers across the US. So in itself, we have the major stakeholders that really participate and acquire new knowledge that GRASP allows.
That's fantastic. Really a fantastic accomplishment. A quick question that was asked by one of our viewers is, how do patients participate in GRASP?
Well, I can start, but I think we'll definitely try to continue the work that we're doing. And initially, the enrollment was in the centers that were participating. We will make more clear, and certainly the SRF will be instrumental to that, to increase awareness of where the centers are active in GRASP are, and this certainly facilitates their enrollment. But I think with publications and with presence in the media, will be easier to understand and to access our network.
That's right. That's great. So if patients are interested to participate, they can reach out to us at the SRF, and we can help
absolutely
connect patients with the GRASP study.
Yeah.
Dr. Gourh, let me ask you a question. Can you talk a little bit about why the genomics piece is sort of the how and why? In other words, how is the genomic information that's generated in GRASP adding to the understanding of scleroderma? And then how does it then lead to potentially new therapies?
Well, thank you, Deanne, for that question, and delighted to be part of this webinar. I think studying the African American population with scleroderma has really helped us identify new genes that we didn't know existed before in causing this disease, whether studying other ancestral patients or studying other autoimmune diseases. One of them example is HLA, which has been shown in a lot of diseases, but now we are finding and trying to understand how the misspellings in these genes of HLA are leading to scleroderma, and not just the disease, but maybe specific organ involvements. And could this be utilized in predicting people who might develop the disease? So maybe you go to the doctor on day one of your visit and you get a blood draw, and we can identify these misspellings in your DNA. And maybe these can be utilized to predict, and maybe you can be monitored early or put in a clinical trial early on. We are also identifying new genes like IFT43, TGA-beta3 locus on chromosome 14, and this gene hasn't been reported before with scleroderma. And I think to piggyback on your earlier question about how do we spread this knowledge? So like Dr. Boin was saying, we have published this work, but we are also disseminating this information at the American College of Rheumatology meetings, which is an annual gathering of all the rheumatologists, not just in the United States, but all around the world. And we've had for last several years disseminated this information. So the idea is that this will not just help advance the science, but also in fact, maybe change the way we are managing patients who are African American in ancestry, but also perhaps of all ancestral backgrounds.
That's fantastic, fantastic. And how does increasing the number of patients enrolled in GRASP aid in the study? And why is it so important?
No, that's a great question again. And as Dr. Kastner was telling earlier, that these studies, we don't think that one gene or one misspelling alone may be causing the disease, but it's an aggregation, the collection of multiple of these misspellings in multiple genes. And to identify more of these genes, we need increased what we call a statistical power. What that means is we need more larger sample sizes. So we can understand all these genes which have a small, minuscule effect size, and we can identify that. So right now, as Dr. Boin had said, we had 24 centers collaborating, and we have over 1,300 patients already enrolled. And if we can increase that number even more, we have the potential to identify newer genes, and that can lead to newer targets for this disease.
If you allow me, Deanne, I want to emphasize that also a uniqueness about GRASP is that we didn't collect information only to say whether the patient has scleroderma or not. We really were granular. These were experts that could really characterize the disease, and this give a lot of power to our scientists to really not only look at the genes, but also every aspect of scleroderma. And that's where the high number is critical. So you can really, intersect the genetic data with all the different clinical features. So a high number allows us to do that in a very specific and unique way that has not done before.
Similarly, I think the collection of blood samples from some of the patients has allowed autoantibody analyses. Is that correct?
Absolutely. And we have the serum of these patients who will totally match also that aspect.
So you're right. So these are newer autoantibodies that are being detected, have been identified in these patients. And we're also looking at trying to identify now HLA genes that have never been previously studied in these patients who have these specific autoantibodies. So you're absolutely right. It's leading to a lot of new work and new discoveries that, again, can identify these new subset of patients with these antibodies and HLA associations.
That's fantastic. Dr. Torok, let me ask you a quick question about the Juvenile Scleroderma Project. So why are juvenile patients so important to study in a disease like scleroderma?
Deanne, thanks for that question, and thanks for allowing me to be part of this group. It's really exciting. Pediatrics scleroderma is interesting because it's even more rare, right? I think it was shown the curve of the onset of scleroderma, and the very, very small percentage, 5% or so, are in those that are less than 15 years old. So those patients are very unique and interesting. So something possibly more genetic is happening to them over the kind of environmental immune kind of turnover that's causing scleroderma. So that's why honing in on those young patients is really important for genetic studies, because they probably have more of a genetic imprint or load. Again, as Dr. Kastner mentioned, it might not be one single gene, it might be a collection of genes, but it might be a collection of a few genes that are kind of driving that. And why we're doing the TRIOS study, which is similar to when Dr. Kastner went all the way over to Israel and got the family's DNA from mom, dad, and the child, et cetera, helps us in pediatrics because the number is so small or fewer. Getting DNA from mom and dad and then comparing that to the child that's affected is helpful, because you take away a lot of variation, because the mom and dad's DNA and you're comparing it to the child. So then you're looking for those variants in the child that's different in the immune system from mom and dad. So that's why the TRIOS kind of approach to the pediatric onset of scleroderma is so important.
Exactly. And so families are very rare in scleroderma, but what would you say to a patient who might be a member of a family that has more than one affected member?
Yes, please enroll them. We would love to get the DNA samples and the clinical information as well. As was mentioned with the GRASP cohort, our cohort collects very detailed clinical information, including, age of onset, et cetera, as well as lung disease, skin disease. So getting those patients enrolled in our study is wonderful. You can contact us either through the Scleroderma Research Foundation or just scleroderma at chp.edu. That I mentioned at the end of the slides. My study coordinator will get back because we do have an open IRB that allows for, grandparents, parents, child, siblings, et cetera, to help give a sample of DNA, and it's just spitting in a little tube. So it's pretty non-invasive.
Wow, that would be super informative. So thank you for that. I have sort of a general question. I don't know who wants to take this one, but I know that something that's always on the minds of patients is how their data is used and how their data is safely and securely stored. So maybe since that's always a big question in genomics, one of you can answer that one.
Well, I'll at least say a few words about that. And those are that the data are kept in very secure servers. They are stored in such a way that they're de-identified in the server, and so that you can't associate a particular name with a particular set of DNA variants. And so there are multiple levels of protection to make sure that your genetic and genomic data would not become general knowledge.
Great. Thank you. I have a question from Valerie about Epstein-Barr virus infection or other viral triggers. And probably people have heard about some of the recent studies in multiple sclerosis. And I think there's a question about whether that could potentially be involved, that a viral trigger could potentially be involved in scleroderma.
Sure, I could take that, Deanne, and I think that's a great observation by Valerie. Recently, it has been shown in multiple sclerosis and other diseases that Epstein-Barr virus may be involved. And I think in some earlier studies a couple of decades ago, there was some invocation of Epstein-Barr virus in scleroderma too. But our earlier work that was published in the Proceedings of National Academy of Sciences a couple of years ago, we are proposing a virus called the Mimi virus that might be triggering scleroderma, at least in a subset of disease, a subset of the patients who develop scleroderma. And we are currently working on that. So it is work from this GRASS project that is developing that will perhaps enlighten us and give us new guidance about whether these viruses could be involved in disease pathogenesis.
Super interesting. So, okay, a general question for all of you. What would you like to say to patients who are considering participating in a genomic study like the ones we're talking about here today?
Well, I would just say, welcome. We would love to talk with you and potentially to obtain samples if that would be appropriate.
And we always said the more, the merrier.
Indeed.
Let's do it is what I would say. Yes, absolutely.
It's an enormous contribution to scleroderma research to participate in these studies, both for individual patients and for the community as a whole. So we really want to thank everybody who's participated so far and encourage others to join us in genomic studies. Let's see. I have maybe one more question here. Which is, if people are thinking of having children after being diagnosed with scleroderma, do they need to worry about scleroderma, or can they can you put their minds at ease a little bit? Anyone?
We have the expert.
Yeah, well, I would say, although I don't know that I'm the expert, but certainly there are so many genes that are usually involved in the susceptibility to scleroderma that in general, this would be something that we would not cause you to think that you should lose any sleep over it. The only possible case would be if one were to discover a single gene cause, which we're still looking for actually in this case. So I think we can set your minds at rest, at least for right now.
Thank you. Thank you. Well, thank you, everyone. This has been such a great session. Thank you for your very enlightening talks and for joining us today. And as always, it's wonderful to see you. So that's it.
Thank you.
Thank you so much.
Thank you.
Have
Deanne.
a wonderful day. Thank you.
Thanks,